Thank you very much. Well, my name is Marcus Bosson. I'm the CEO and co-founder of QuiaPEG Pharmaceutical, a biotech company based on a proprietary drug delivery platform. We are located in Uppsala, former Pharmacia area. We have our lab and offices there. So, our drug delivery platform is called UniCleaver. It's chemistry combined with polyethylene glycol, and I will come back to you in a minute what exactly we do. So, we are based on a scalable business model. So, we usually say we are not a one-trick pony, one project, and we kind of stuck to that project. We have a quite broad platform, which is scalable. So in the future, we can actually get short-term revenues in form of licensing of a drug delivery platform to pharmaceutical companies developing new pharmaceuticals where the half-life is a problem. We could extend that half-life. And in parallel, we are working on building our own pipeline of biobetters, utilizing already approved drugs and making it a longer half-life. That means instead of once daily, maybe once weekly. That's what we do. So, is there any money in this kind of business with drug delivery and half-life extension? Well, we have a benchmark company. It's called Ascendis. It's based on German invention, Danish entrepreneurship, and excellent business craftsmanship. And it's listed on NASDAQ in New York City. And actually, I checked the market cap today. It was founded 2008, and it's even $7 billion. So it's one of the top 100 companies, biggest pharma companies in the world today. And they have a similar technology, increasing the half-life with a releasable technology platform. And they have succeeded now in a phase 3 clinical trial with human growth hormone, where instead of once daily, you take it once weekly. So definitely it's interesting. As you can see, they did some years ago out-licensing to Sanofi, Genentech, and United Therapeutics, and it totals up to about 60 million euros. And that was preclinical, only for the platform, nothing regarding specific projects. So, UniCleaver, how does it work? Well, as I mentioned, it's a prodrug. And what does a prodrug mean? It means when you couple our technology to a molecule, a peptide protein or a small molecule, it kind of protects this drug. And then it's released by pH. We all have the same pH, 7.4. So depending on how you design this activated PEG, which is proprietary, you can have different cleavage time. So, for instance, 50 hours or 10 hours or 80 hours, depending on the substance. It protects, it's biologically inactive. And then with the pH, it starts cleaving. And once it has cleaved, it's fully native, the biological effect comes back. And polyethylene glycol, PEG, maybe you have heard of a golf PEG, but this is chemistry. It is widely used, has been used for 30 years. You have several blockbusters utilizing this PEG, Cimzia, Neulasta, etc. Basically, literally, millions of people are using PEGylated drugs every day. You have all used PEG today, either by creams or food or something. It's widely used. So that means it doesn't have side effects, potential side effects, but it has been tested. So it's well known how it works, pros and cons. So what you then take is the drug, and then we have our proprietary technology called UniCleaver. So you connect it together. And then you can design this UniCleaver, depending on the half-life, how long it circulates in the blood, a different trigger from this library. I will come to that picture. And then when pH kicks in, you get the split, the cleavage. So you have a PEG for itself, and it's cleared through the system, and you have an active drug which can do the job. So this is a summary of our invention. So it's in the middle you have a phosphotriester with a trigger, which you select from this trigger library, where you have different cleavage time. So you can tailor-made this release speed When you have a polyethylene glycol and the drug. So by combining this, you can tailor-made the half-life of the drug, increase the half-life of the drug. And we usually say it's universal. It's triggered cleavage by either enzymes or pH. And enzymes could be very important in the development of antibody-drug conjugates, which is a big area in cancer oncology today. It's efficient, so you have a complete cleavage. You don't have any residue left on the pharmaceutical, which is good from a regulatory perspective. It's predictable, it's controlled, and we have a growing patent estate. And now I see that actually BioStock used my old presentation, so I have an updated version, unfortunately, but I will do my best. So, and the clinical advantages is actually fewer doses, increased patient benefits, and lower cost to society. With stable blood concentrations, you can most likely reach a better, less adverse effects. And regulatory-wise, you can use this called the pathway called Section 505, which is you can piggyback on what's already out there in the filings, documentations of original pharmaceutical product. So we call the biobetters. Why is it biobetter? You have all heard about biosimilars. Biosimilars, that's supposed to be similar to the original drug. When it's small molecules, you call it generics. In bio, you call it biosimilars. Biobetters, that's supposed to be better than the original product and the biosimilar, which means that you add something good for the patient. That means you can maybe patent it also, protect it. You can maybe also have a flexibility of getting a premium price instead of competing with the lowest price possible. And also you have some other goodies, data exclusivity, etc. So we selected some time back how to show this, all what we're saying, how to show that this is really true. So we went through different potential pharmaceuticals, and we actually came up with this as a model project, pegylated liraglutide. And it's a peptide, and it's a blockbuster drug for Novo Nordisk. The patent is soon expiring. It's about $4 billion annually. It's administered daily by injection, and also a key thing is that 10–15% of the patients, they stop taking it because of side effects, nausea, vomiting, not feeling well, to put it simple. So what we are trying to do is actually to develop a biobetter, to be administered once weekly, and potentially with less side effects compared to the original. And we have filed a patent on this, so that expires 2038. More patents will follow regarding production, etc., so we can probably extend it. And it's also a possibility to file this under Section 505(b)(2). And what's really good about the potential of less side effects is that, as I said, we have this protection of the original drug and its controlled release. That means we don't have that spike when you inject it the first time. So you could have this more steady state of release of liraglutide or any other peptide, which means that potentially it could be less side effects, and that's very good sales argument. So unfortunately, I had an updated version, but that doesn't seem. So I will go over to the pharmacokinetic pilot study. We actually had two press releases. One was last week with that we had finished the development of two analytical methods. It was together with a global CRO, very well renowned. It has taken some time, so we made one for liraglutide and one for the conjugate, the pegylated liraglutide. And we need that to measure the different levels moving forward also in preclinic and eventually clinical trial. And then we released a couple of days ago the press release of a pharmacokinetic pilot study. So we had a rat study. They were injected subcutaneously and intravenously. Subcutaneously means under the skin, and IV means into the blood. And the advantage with sub-Q administration, subcutaneous administration, is that you don't have to go to the doctor to administer it. So we measured the pegylated liraglutide, and then we had one control group with liraglutide. And what we see is actually the liraglutide is released as expected from the conjugate. It reaches the bloodstream when injected subcutaneously, and for you, well, it doesn't sound much, but it's actually an important milestone to tick that box that we could inject it subcutaneously and get this controlled release. It's actually a milestone. And after the administration, we can see that the plasma half-life, that the half-life of this in the blood is significantly extended compared to direct administration of liraglutide. So we're really onto something here. And this not only validates this project so far, it also validates the Unicleaver technology platform, that if it works with this peptide, maybe it also works with, supposedly, with other peptides and proteins. So it's really very good data. All right, I will quickly go through the company, the management, and I'm the co-founder, and Marek Kwiatkowski co-founder, and we have a board with a chairman, Steen Kreuer, who used to be the chairman of the CEO of AstraZeneca in Shanghai, and actually built it through 6 years and made it to what it is today. We have a growing patent estate. We have 11 approved patents. We also have a good shareholder structure. Neuenburg Investment Partners, which recently, I think, joined Episurf shareholder community. And also, you can see that the founders, we have a lot of skin in the game, and I think that's one important signal to you as potential shareholders or shareholders, that we are really dedicated financially also to this company. So the ongoing and planned activities, we have announced that we will soon have proof of concept results, and that's ongoing. And those results will be very, very important for us. It's a big study. We have pegylated liraglutide, 2 doses. We compare it, benchmark it to liraglutide 2 doses and to the semaglutide 2 doses in which is Novo Nordisk once weekly peptide, which also have the same adverse effects as liraglutide. So, and then we have a control group. So it's a food intake study. So you have rats, they're running around, and the control group, which has not received anything, they eat and are happy they eat during the night, actually. So for a certain amount of time, you measure how much they eat, and also you weigh them once daily. And they are supposed to, with this liraglutide, pegylated liraglutide and semaglutide, to lose weight compared to the control. We'll see what the results will turn out to be, but it's really a proof of concept study for us. And that's going to be announced soon. In parallel, we have a CMC aspect ongoing, and I agree with a previous speaker that contract manufacturing or manufacturing is actually one of the key things for every biotech company. If you have any delays, if you have any cost increase, it's due to that, I can almost guarantee you. So that's ongoing. We have a very good partner in Germany. It's kind of a firm where to go to when it's pegylation. We have planned now additional preclinical studies and also the tox studies. And last but not least, we have ongoing serious licensing discussions in China with a potential partner for this pegylated liraglutide. There is a big interest in China for GLP-1, which is diabetes and oncology, and new technology platforms. We have been to several conferences. It's huge interest, and they do have money. So, but they don't have exactly that kind of technology. So Now they are focused to new inventions. And also, we have, as you know, some ongoing collaborations, testing of this Unicleaver platform. That's progressing, and we also have other ongoing discussions. And with these data coming out now, I'm pretty sure that we will enter a very important phase in those discussions. So that's where we are with the business development. And let me just finalize why invest in QuiaPEG. Well, Unicleaver is a versatile platform, limited competition. I usually say it's about 5 worldwide companies, and a couple of those, they don't out-license their technology. So I think we have a good chance of, if we reach that level of publications, awareness of what we do and who we are, I'm pretty sure we do have a very important position in this market. And parallel, we are value creating by building a pipeline of biobetters. That means limited risk for the investor because it's known pharmaceutical drugs. And we have this also reduced commercial risk because it's really accepted the pegylation area, utilizing pegylation as a sort of a form of half-life extension. And last, it could be also an interesting potential acquisition target for a big pharma company. As I mentioned before, we also have technology for ADCs, the linker technology, and we have this for biobetters and also for new molecules. So a big pharma with a big pipeline, they could really make use of this platform. And by that, I thank you for your kind attention.
Thank you very much, Marcus. Sorry about the mix-up with the presentation.
Okay.
I think you got a fairly interesting message through anyways.
Okay.
So something that caught my interest was this, when you work with creating a biobetter, you said that there's a low risk and it's quicker than to develop a brand new drug. And but what are the differences between to develop a biosimilar towards a biobetter? Is it not a bit longer process?
I think, yeah, it's a biobetter because you insert something which makes it better. So of course you have regulatory-wise a little bit more burden than a biosimilar, which is supposed to be about the same, no better, no worse than the original drug. So, but I mean that in that field you have plenty of big competition. Big is beautiful, and then you need to have a marketing setup and be aggressive and put a lot of time and money into marketing. Biobetter, we achieve something which is better, which means that if you have original product up here, price-wise, you have maybe, and then after it comes off patent, it will come down, but often, like Novo Nordisk, they have a semaglutide, which is still up here, price-wise, but the liraglutide might be down here. And because we will be compared to the semaglutide, which is the once weekly, we have a flexibility in between to put us price-wise. So we don't have to compete with the lowest price possible. So that, but yes, it's a little bit more work and a little bit more investment, but I think in the long run, when you don't have to compete with the price, you can make it back quickly. So.
And then looking from the other perspective, then working towards finding a biobetter for one of those, well, blockbusters, really. The original owners, like Novo Nordisk, for example, they should be monitoring your activities fairly closely, I would say. Are they possible takers for your project?
That's speculation, but
of course.
yeah, of course. But maybe the big ones, you know, they have not invented here attitude. I don't know. I don't know how familiar just specifically Novo Nordisk is, but it might be so. So, but you have several others, of course, to take us to this. So, and I should point out that maybe we will take this through not only a tox, but maybe a phase 1/2 2A, so we can measure the efficacy in humans, because that would be tremendous value for all other potential targets we have. So.
Yeah, and about that, it seems like this platform technology, if you will, has a broad potential. And are there any? You mentioned that a potential acquisition target, sitting on an asset like this, what would make you take a decision that you would actually let someone buy you when you have that potential?
I don't know. It's a
huge bags of money. Or
yeah, well, you can say today. I don't know what is it, 100 million Swedish krona, give or take a few, and that's $10 million. And if you ask yourself, $10 million, how much is that for a big pharma company? Maybe not so much. But I'm not speculating about the stock price. But I can see that in the future, this really now when we are gathering hard facts that this could be really a good target for acquisition and a clean cut. And I expect us, a co-founder, put a lot of time and effort and money into it. I expect a lot more. So,
are there any questions from the audience? We do have a question. There you go.
Just what is the present patent situation for liraglutide?
Liraglutide expires, I think I had it on a slide, 2022, 2023, and then you have some production patents, so you have a tail of expirations.
So in practice, when can you launch without running into problems?
That will not be a problem as far as we see it, and also I should point out that we will Novo uses it in bacteria production, and they have a lot of patents regarding that. That's the problem for the biosimilar manufacturers. So the biosimilars are making it in synthetically, and we will use a synthetically produced liraglutide. It's very important to point that out, and that opens also the doors for us to, if they have already, the biosimilar manufacturers, taking that investment into a process for making synthetic liraglutide. I mean, the additional investment to make something together with us, it's not that great. So that's a good sales argument when we are talking to potential partners for this.
Well, Marcus, thank you very much.
Thank you.
Time's up. You can, of course, be available also to answer questions after the presentation.
Yes, I will. Yeah. Okay.
Thank you very much.
Thank you.